Okay, hello everyone. So this is the week 5 question answer session of the NPTEL course Fundamentals of Protein Chemistry. This course is coordinated by Professor Dasgupta from the Department of Chemistry, IIT Kharagpur, and I am Beas Mukherjee, a Prime Minister's Research Fellow from the Department of Bioscience and Biotechnology under the supervision of Dr. Shomode. And today in this session we will be discussing some topics of protein ligand interactions, the different techniques that are used for studying protein ligand interactions, and we will also talk about allostery and different kind of regulations in proteins that are mediated by interactions with ligands. And you can ask me your doubts and questions. We will also be solving related questions. Okay, so we'll go through a little bit of theory and then solve a few questions. This is how we are going to go about with today's class. Okay, so as the students start joining, we will begin with the class and they will join in the meantime. Okay, so firstly let's talk about protein ligand interactions. Protein ligand interactions refer to the binding interactions between a protein molecule and a small molecule, that is a ligand. So in biological system, proteins often interact with various ligands, which include small molecules, ions, drugs and other proteins. So these interactions play a very important role in the numerous biological processes, including signal transduction, enzymatic reactions, transport of molecules across cell membranes and regulation of gene expression. Now, the binding of a ligand to a protein typically occurs at specific sites on the protein surface. These are called the binding site or the active sites. Right. So now these binding sites are complementary to the shape and size and the chemical properties of the ligand, allowing for very specific recognition and binding. Now, the binding process is often characterized by high affinity and specificity with the protein. And the binding can be weak binding, it can be tight binding, so it can be in micromolar, it can be in millimolar range, it can be in nanomolar range. Hello? So, in today's class, we are discussing protein ligand interactions. Okay? Let's go about with the class. So this slide, you guys are already familiar with this one. So you know how proteins interact with ligands. There can be different kinds of interactions. For example, there can be hydrogen bonding interactions, ionic bonds and hydrophobic interactions, van der Waals interactions, etc. Right? So these are the different interactions using which the ligands bind to proteins. Okay, so we will start with a small and simple question. Okay, so based on your understanding, you can answer this question. Now you already know the drill. You have to type in the chat box, okay, or you can unmute yourself and you can give me the answer to this question. All right. So the question is, the binding surface of a DNA binding protein is likely to be negative, positive, neutral, and it cannot be predicted. So which one do you think would be the right answer? Right, so you have to think about the charge of the DNA and you have to understand that this DNA binding surface of the protein should be able to attract the DNA towards itself, and so the binding surface should be of what charge, right? So that is something that you have to answer. Now, one very interesting thing I can tell you regarding this, since in our lab we also work with DNA binding proteins, okay, and RNA binding proteins. So now, interestingly, if you look at it this way, that normally people would say that, okay, if this is, let's say the DNA is having a particular charge, the protein is having, let's say, the opposite charge, it is going to bind. But then you also know about DNA binding specificity of transcription factors, right? Now, transcription factors, they interact with the major groove of DNA as well as minor groove of DNA. Okay, so when they are interacting with the major groove of DNA, that time it is recognizing the nucleotides that are there in the major groove. So that time it is a base specific interaction. That means each each nucleotide or each nucleotide nitrogen as base is being recognized by the residues and the chemical groups of the residues, and then there is binding between these, right? So that is base specific binding. And when this transcription factor, regions in the transcription factor bind to the minor groove in DNA. The minor groove is very narrow. It is very difficult for regions in the protein to enter into the minor groove. So these are often these intrinsically disordered, very flexible regions in the protein. Okay. Now these bind to the minor groove based on shape specificity. So that means the minor groove is very narrow and it's like this in the shape. So the protein is going to bind this way, and it's going to be very charge specific majorly, right? So now that is something new that I've told you. So you can answer this question. It's a simple question. You can answer this. Binding surface of a DNA binding protein is likely to be of what charge? Or is it going to be neutral? Is it not predictable? Tell me. Okay, we'll look at the answer. Right, so it is actually going to be more positive because DNA is negative in charge, right? The phosphate backbone is there, and that is why you are having this negative charge in DNA. And so the binding region should be positively charged even if the entire protein is having different charge. So that is a different matter altogether. But the binding surface that is going to interact with the DNA, this is majorly made up of basic residues. Largely arginine, lysine, all these residues are majorly present so that these can help in binding with the DNA. Okay. Right. Now, there is something called allostery. Now, you guys have already read about allostery earlier, right? So, what is allostery? Allostery is that there is a particular protein ligand interaction. Now, because of this protein ligand interaction, let's say there is some modification in the protein. It can be change in the conformation of the protein. It can be change in activity of the protein, and this allostery is actually affecting the overall function of the protein. So this is a form of regulation of the protein. Okay. So for example, now there are enzymes that are not regulated by allosteric interactions, and there are enzymes and other proteins that are regulated by allosteric interactions. So there is this very subtle difference. So see, in an allosteric enzyme, activity in the allosteric enzyme, the activity or function changes with respect to binding of a regulatory molecule at a site other than the active site. So now at the active site, the substrate binds, right, for this enzyme. But at a site other than the active site, you can look at this figure. So there is the allosteric site where this allosteric molecule goes and binds. Now this regulatory site known as the allosteric site is distinct from the active site where the substrate binds. Binding of a regulatory molecule to the allosteric site induces a conformational change in the enzyme structure which either enhances the activity of the enzyme or it reduces the enzyme's catalytic activity. Okay, and another thing is if you look at these graphs, so with increasing concentration of the substrate, the activity of the enzyme initially increases and then it gets saturated, and so it is undergoing the saturation phase, right? So this is a hyperbolic curve. But when there is allostery, initially it is going to rapidly increase and then it undergoes this saturation phase. So this is this S-shaped or sigmoidal curve. So one is hyperbolic, the other one is sigmoidal. So these are very important questions that you can expect in your exams. Okay. Right. So now based on this that we have learned until now, you can try to answer this particular question. So which one is true regarding an allosteric interaction? A, the binding of a molecule to a binding site inhibits the binding of another molecule to the same binding site. The second one is, so which is true? Okay, so binding of a molecule to the binding site affects binding at another site. A binding of a molecule to a binding site affects binding at a similar site at another monomer. Multiple molecules compete to bind at the same binding site. So which of these are true? for an allosteric interaction. One of the points is very, very direct. Okay, B. All right, let's see. Yes, that's right. B is the right answer. So if we go back to our figure, right? So if we go back to this particular figure, you will see that the substrate binding site is a separate site. This is the active site, whereas your allosteric effector molecule, this is binding to the binding site, regulatory allosteric site. So yes, that is correct. This is B is the right answer. Now, how else can you try to understand this? So, see, multiple molecules compete to bind at the same binding site. Now, if you look at the perspective of inhibitors, so then you will realize that for competitive inhibitor, multiple molecules, they are binding at the same binding site, and that is the inhibitor and the substrate. These are both binding to the active site. Right? So, this is how, if you look at the other points, they will actually get eliminated. Okay, now we have another question. So, which of the following is or are correct concerning cooperative binding of a ligand to a protein? Now, what is cooperative binding? So, cooperative binding is a feature of allosteric interaction. This is largely seen in allostery regulated molecules, where what happens is, due to when, let's say, a particular molecule is supposed to bind to something, and let's say that this particular allosteric effector molecule is going and binding to it, and this other molecule is also coming and binding to it. Then what happens is, it actually affects the other monomers in that quaternary or tertiary structure, to bind to the molecule more. So what happens is, its activity increases drastically. So that is cooperative binding. It is seen in hemoglobin. Okay. Now, which of the following is or are correct concerning cooperative binding of a ligand to a protein? One, it is usually a form of allosteric interaction. It is usually associated with proteins with multiple subunits. It rarely occurs in enzymes. It results in a non-linear hill plot. So let's see which of these are correct. So you're saying that it is A. A, 1 and 2. Okay, let's see. Okay. Okay, so C is actually the correct answer. So 1, 2 and 3, all of these are correct. Okay, let's see. Let's have a look at that. So one of the examples for this is the regulation of hemoglobin. Okay, so there's something known as Bohr's effect. Now these are very important for your exam point of view. Okay, so you can go through this, how hemoglobin is getting affected because of the different pH condition, different temperature condition, different oxygen levels, different CO2 level, etc. Okay, so Bohr's effect refers to the phenomena in which changes in the concentration of hydrogen ion, that is the pH change, and carbon dioxide change affects the oxygen binding affinity of hemoglobin. Okay, so the Haldane effect describes how this binding of oxygen to hemoglobin affects the hemoglobin's ability to carry carbon dioxide and H plus ions. So the Bohr-Haldane effect describes CO2, sorry, this shouldn't be H plus ion, this should be O2, okay, in the sentence above, okay. So carbon dioxide and oxygen, okay. So in the Bohr-Haldane effect, carbon dioxide, one second, I'll just fix this because otherwise there will be confusion regarding this. Yeah, okay, now we'll carry on. Okay, right. So see what happens in Bohr-Haldane effect. This describes how carbon dioxide and oxygen is carried by erythrocytes and how that is influenced in changes with respect to changes in the pH condition. Okay, so more acidic environment increases the onloading of carbon dioxide and offloading, that is release of oxygen. So these effects work opposite to each other. So Bohr says, what is Bohr effect? So Bohr effect tells you that offloading of carbon dioxide at the lungs, because carbon dioxide needs to be released, right? Loading of loading of oxygen onto hemoglobin. So from lungs, fresh oxygen is loaded on into the hemoglobin of RBC and carbon dioxide is released. And the presence of oxygenated hemoglobin shifts the oxygen dissociation curve towards right. Then there is the Haldane effect. So Haldane effect tells you loading of carbon dioxide from the tissues. So the tissues are constantly metabolizing, and due to respiration they are giving out carbon dioxide, right? So now what happens is this is collected from the tissues, that is Haldane effect, and offloading of the oxygen from hemoglobin. So the presence of deoxygenated hemoglobin shifts the carbon dioxide dissociation curve to the left. Okay, so we will look at this in more details. Now, oxygen regulation of hemoglobin. So you see, there is this. Now hemoglobin exists in two different states. One is the relaxed state and one is this tight binding state. Okay, so there is this R state and then there is this other state, right? T state, which is the tense state. Now see, oxygen binding and unloading. So oxyhemoglobin. Now in oxyhemoglobin, what happens is as the pH increases and partial pressure of carbon dioxide also increases. So increase in 2,3-DPG. So this is bisphosphoglycerate, right? Bisphospho or diphosphoglycerate, which is an important allosteric regulator. You have come across this molecule when you learned about glycolysis. So this is also there in glycolysis, right? So now falling partial pressure of oxygen. Then what happens? This carbon dioxide changes from oxyhemoglobin to deoxyhemoglobin, and it goes from this relaxed binding state to the tight binding state. And again, when the partial pressure of oxygen increases and carbon monoxide is there, then it goes from tight binding state to relaxed binding state. Right? So the presence of oxygen stabilizes the R state. When two oxygen molecules are bound, the R state predominates. Okay, and the curve of hemoglobin, the percentage of oxygen saturation curve, this is actually a sigmoidal hemoglobin O2 binding curve, is essentially the sum of two hyperbolic curves, right? So this hyperbolic curve is actually the theoretical pure R state curve and theoretical pure T state curve. So because of these two curves, you get the general sigmoidal curve of the actual hemoglobin. Now, why is it sigmoidal? Because this is also regulated by allosteric interaction. Now, which are the allosteric effector molecules? All of these different things, especially the 2,3-DPG. These are the different allosteric regulators for hemoglobin. Okay. Right. Now we will move on to the next one. So see, this is given in more details. Now what happens is, as the pH increases and the level of bisphosphoglycerate decreases and temperature decreases, then what happens? Then shift in the curve is seen, and also that is actually Haldane effect. So in Haldane effect, O2 displaces carbon dioxide from hemoglobin, and in Bohr's effect, the carbon dioxide concentration increases and there is decrease in the pH and increase in DPG and increase in temperature results in Bohr's effect. So these are the two things that you have to understand. And there is shift in the curve. So shifting in the curve can be observed. Right. Now you can try to answer this question. So which one is a false statement? A says 2,3-BPG allosterically binds with the beta chain of hemoglobin in the central compartment. Try to recall the figure that we just saw. Okay, and 2,3-BPG helps to release oxygen from hemoglobin even at low partial pressure of oxygen. 2,3-BPG increases affinity of oxygen of hemoglobin, and the deoxygenated form of hemoglobin is stabilized by 2,3-BPG. So which one do you think is the right answer? Which statement is false? Okay, we'll look at the right answer. Okay, so the false statement here is C. So C says that 2-BPG increases oxygen's affinity of hemoglobin. So actually 2,3-BPG decreases the oxygen affinity of hemoglobin and facilitates oxygen transfer from hemoglobin to myoglobin at the tissues. So if we go back to the slide, we will see that due to this 2,3-BPG, this actually binds between the two beta subunits of hemoglobin, and it is stabilizing the deoxyhemoglobin T state of hemoglobin, and 2,3-BPG concentration as it increases, it is ensuring that oxygen is released from hemoglobin. Okay, and that is why all the other statements are correct, but the third one is not correct. Okay, now we go to something known as the Scatchard plot. Now whenever we are talking about protein ligand interactions, how do we quantify protein ligand interactions? That is done using Scatchard plot. Now Scatchard plot is very important, so you have to understand this. It is a very simple concept but very important for your exams also. Okay, now Scatchard plot is a graphical method to visualize the binding characteristics of a ligand to a protein. It plots the ratio of bound ligand to free ligand against the amount of bound protein. Okay, now so the Scatchard equation is given by bound concentration of the bound form divided by the L, that is concentration of free ligand, equals to minus amount of ligand bound to the protein divided by Kd. Kd is the dissociation constant. Okay, plus the bound max. So bound max is maximum amount of ligand that can bind. So let's say there are a protein has two ligand binding sites. Now there will be a lot of protein molecules and there will be a lot of ligands. Now at a time it is very it is possible that both the sites in the protein will be occupied by two ligand molecules when the concentration of the ligand is, let's say, very high, right? But sometimes both the sites are not occupied by the ligands, and so that is called the total binding site. Okay, when maximum capacity of the protein to bind to the ligand, that is taken into consideration. Now Kd is the dissociation constant. So in a typical Scatchard plot, if the data points form a straight line, it suggests that the binding sites are equivalent. That is, the ligand binds to a single class of sites within the same affinity. Okay, and the graph is plotted for concentration of bound form divided by free form, and in the x-axis it is bound form. Now slope is minus k and minus k or minus kd, and Bmax can be got from this axis. Okay, now curvature. If the plot shows curvature, it indicates, now if the plot is just straight, that tells you that the ligand binding sites are uniform. There is only one kind of ligand binding site. But if there is some kind of curvature, it indicates that there might be multiple classes of binding site with different affinity or complex interactions. So that is something that we can come to know. Now these Scatchard plots are very important for any kind of protein ligand interactions. Okay, now we will try to look at these Scatchard plots. Okay, So now see, A and B correspond to the slope and x-axis intercept respectively of the Scatchard plot. Now option A and B. So you have to tell me what is A and B. Okay. So A and B corresponds to the slope and x-axis intercept. I think this is the answer slide. Okay, anyway, you try to answer. So KD, binding stoichiometry, KD, rate of dissociation, KD, the fractional occupation of the sites, and binding stoichiometry, and B is the KD value. So A and B, x-axis intercept and the slope. Okay. Try to answer. Now, based on whatever is there in the figure, you can try to answer. Right? So, there can be different ways of representing the same thing. See, there can be this Scatchard plot where the y and x-axis values are different. So, that means you're plotting bound fraction by free fraction in the y-axis and bound fraction in the x-axis. For titration curve, it goes this way. So titration curve means bound divided by log of concentration of the free fraction, and for hill plot, it is log of this V by 1 by V and log of L, that is the free form. Okay, so you can read about all of these and you can find out. So we'll look at the answer. The answer is A. Now see, now these are all questions from the previous years, NPTEL, same course, okay. So that is why I have included these. So the more of these kind of questions you practice, better understanding you will have. So this V by L versus B gives a straight line with a slope Ka according to the equation V by L is equal to N into Ka minus V by Ka. So at x-intercept, V by L is equal to 0. That is N Ka is equal to V into Ka, or N is equal to V, and the x-axis intercept indicates stoichiometry. So from where is it coming? If you go back to this, you will understand that all of this is explained here in the Scatchard plot. So whatever we studied right now, V and L, so it's all just terms in the Scatchard plot. So all you need to do is just go through this and you'll be able to understand, right? So here it is explained very nicely. So see, let's say that you want to understand this in terms of equilibrium, okay, of the reaction. So initially there is this protein ligand complex or protein and ligand, and it is forming a complex protein ligand, okay, and the rate of formation, so there is K off and K on. So K off and K on means how easily the two molecules are breaking up, that is the off rate, and how easily they are forming, that is the K on. So dissociation constant tells you the concentration at which half of the ligand binding sites are occupied. So concentration at which half of the ligand binding sites are occupied, that is the dissociation constant, and that is the concentration of the free protein and ligand divided by complex. So that is K inverse divided by K1. Okay, that is off rate divided by the on rate. And K association and K dissociation are inversely proportional. Now, if you look at a reaction at any point, then you know that the reaction quotient is equal to this particular formula, right? And equilibrium constant means the exact same thing but at equilibrium. So, the concentration of all the different entities will be recorded at equilibrium. Now you know about the thermodynamic equation delta G is equal to delta G naught plus RT ln Q. Now instead of Q, if you put K, that is the equilibrium constant, then you get that delta G is 0 and delta G is 0, so that means it is equilibrium, and Q is equal to KEQ. So these are the two conditions for equilibrium, and then we get delta G naught is equal to minus RT ln Q. Okay. Now if you remove this ln with log, then you get this value KEQ is equal to 10 to the power minus delta G naught divided by 2.303 RT. So dissociation constant, if it is very high, that means there is weak binding. If it is very low, that means there is very strong binding. Okay, higher affinity. And the units of dissociation constant and association constant are also mentioned in the slide for your understanding. Now you try to answer this question. For a tight binding, which one is true? So K equilibrium, should it be much less than 1? So should it be negative? And KD, should it be more than 1? So, and which one do you think is the right one? So K equilibrium and KD relation for tight binding. Now when dissociation constant is written with respect to thermodynamics, then this way D is written. Okay. Okay. So you are saying that. Second. Okay. So see, the right answer here is actually D. So tight binding indicates that thermodynamically very stable binding is happening. So delta G is much less than 0 because it is a spontaneous reaction, right? And delta G is equal to minus RT ln KEQ, where delta G is much less than 0. So that means both of these are negative. Both the terms will be negative, and so the KEQ will be much more than 1. Okay, and KD, you know that it is equal to this P into L divided by PL, and the dissociation constant will be small for tight binding. Therefore, KD will be much less than 1. So KD is much less than 1 and KQ is much more than 1. That gives you very tight binding. Okay. Okay. Now we go to the next question. So which among the following is or are true about the thermodynamics of protein ligand binding? So when KD is very low, delta G of binding is very high. Now which is true? Okay. Almost all binding sites are saturated if the ligand concentration is 10 times the KD. The unit of KD for a 1:1 protein and ligand is L per mole. Liters per mole, this is what they're saying. Okay, and KD includes only an enthalpy factor but no entropy factor. So which is true? That is what they're asking. C. Okay, let's look at the right answer. Okay, so the right answer here is B. Now why is that so? Because when binding is very strong, that is delta G is negative, right? So of course delta G value cannot be very high, it would be negative. So 1 would be wrong. Now 1 second. Okay, 1 second. I might have to confirm the answer for this one. So when binding is very strong, that is delta G is highly negative, dissociation is also very less, and KD is also very small. Okay. Now when concentration of the ligand is much more than the concentration of the protein, then nearly all the ligands are bound. Now KD is obtained from halfway of binding, and in that case the concentration of ligand is much more than the Kd. So the ligand concentration is much more than 10 times Kd, right? And for 1:1 complexation, the unit of Kd is P into L divided by PL. That is, now all of these are molar, right? So molar divided by molar. So that is molar. Molar is the unit of Kd, and Kd includes both enthalpy and entropy factors. So 4 would not be the right answer, and 3 would also not be the right answer because here the unit is wrong. So Kd is in molar. Okay, so that is mole per liter. Liter per mole is wrong. It's mole per liter. If it would be association constant, then it would be different. Okay, right. So then 3 is wrong, 4 is wrong, and you are left with 1 and 2. Because this statement is correct. The second statement is correct, and first one, the explanation is given here. Okay. Now this is a numerical. It's a numerical that they have given you. So delta G naught and delta S naught calculated from the following data are so and so. Okay. Now they are so the temperature in Kelvin is given and KEQ is given with respect to 10 to the power 5. Okay, so whatever value is there into 10 to the power 5, and then now they're telling you to find out delta G naught and delta S naught. So how are you going to find? You know the equation, right? Based on the equation, you're going to find out delta S naught and delta G naught. And these kind of questions are common for numerical aptitude test in GATE exams. So it's advisable if you practice the questions, especially numerical questions. Okay. So we can look at the answer. So this is how you're going to solve it. So all the different values are given. Based on that, you get this graph, and from there you get the slope, and you get this change in enthalpy, that is delta H naught. You're getting as this particular value, and delta S naught will be intercept delta S naught divided by R, and from there you get, you know, the value of R, and so this is the particular value that you're getting for delta S naught. Now delta G is equal to change in enthalpy minus temperature into change in entropy. Right? So if you put that into the equation, you get 35.5 kilojoules per mole. So then your right answer would be which one? 33.5 kilojoules per mole. This is for G, and delta S naught is 8.4 joules per mole per Kelvin. So this is 8.4 joules per mole per Kelvin and 33.5, and it's positive. So this C would be the right answer. Okay. Okay. Now enough with the numericals. We are going to discuss some. One second. So there was some connection issue. Now we will talk about protein-ligand interactions and the different techniques that are used for studying protein-ligand interactions. Okay. Now one of the very common techniques is surface plasmon resonance. Now there are different techniques. I'll just list them out. So one is surface plasmon resonance. Then there is ITC, that is isothermal calorimetry, and then there is you can do EMSA if it is protein DNA interactions. Quantification, you can go for electrophoretic mobility shift assay, you can go for fluorimetric techniques, and there are different kinds of techniques like this for quantifying protein ligand interactions. Now depending upon the ligand you can choose the different techniques. For example, for ITC and for, let's say, so the ones where you need lot of quantity of the samples like ITC, because of the heat the samples are destroyed, so you cannot reuse it. Okay, and for, let's say, protein DNA interactions, protein RNA interactions, you have EMSA and other fluorimetric techniques. Okay, so we'll start with surface plasmon resonance. Now plasmon refers to electrons. Okay, and these are special electrons that are emitting from this gold surface. Okay, so see what happens here. So SPR is a label free technique. So for this you do not need to label your protein of interest or let's say ligand or DNA of interest, anything. So there does not need to be any radioactive or fluorescent label. Okay, so label free technique to study biomolecular interactions in real time. And in SPR, one binding partner, typically a protein, is immobilized on a sensor surface, and the sensor surface is often gold. So there is a surface while the other binding partner, that is a ligand, it is flowing through it. Okay, and changes in the refractive index. Now light is coming and falling onto it. Now without this immobilization you are getting one kind of reflection, right, reflected light. Then with immobilization you are getting a different kind of reflection angle. Now when the ligand molecule is binding to the immobilized protein, you are getting different reflection. So based on that you are going to find out the different characteristics of binding. So changes in the refractive index at the sensor surface due to complex formation is monitored, allowing for the determination of binding kinetics, affinity and specificity. All of these three can be figured out. Okay, so objective. Now surface plasmon resonance helps perform molecular interactions in real time. So real time means that as the interaction is happening you are monitoring it. Okay, now the studies can be used to observe if two or more interactants bind to each other. It can also be used to measure how strong the interactions are and to even measure the actual association and dissociation rate. So K on and K off, that also you can find. Specificity, affinity, etc. Okay, so the objectives of this experiment are firstly to perform protein-protein interaction using SPR, to analyze the molecular interaction obtained as an SPR curve or a sensogram. So all the data that you are getting because of this interaction, this you are getting in the form of a sensogram. For example, in FPLC and in ion exchange chromatography and all, we studied that there was this chromatogram, right? Because that was a chromatographic technique. Now this here you get it as a sensogram. Now to calculate the binding affinity of the protein interactions performed. Okay, so techniques to study protein ligand interactions, and we are at SPR. SPR is a phenomena, surface plasmon resonance is a phenomena that can be observed in a thin conducting film at an interface between media of different refractive index. In SPR system, the media are made up of glass, sensor chip and the sample solution, and the conducting film is a thin layer of gold on the sensor chip surface. And SPR is a physical process that can occur when the plane polarized light. So plane polarized light means that the electric field and magnetic field vectors in light, they are oscillating in the same plane. So that is a plane polarized light. This plane polarized light comes and hits the thin metal surface and total internal reflection is happening. So if you look at the figure, you realize that this is the incident light. It is falling on the surface of the prism and it is getting totally internally reflected, right? So entire light is coming in. So that's total internal reflection. And you see that when it is passing, it is going from a denser to rarer medium, and that is why this total internal reflection is happening. It is more than the IC. Now see, changing the angle of incidence. Why plane polarized light is required? Okay, so why is plane polarized light required? Because why is it required? Because if the light is not plane polarized, then I will have to look into it. You also search. Okay, it's a very good question, and we can figure that out together. Okay, all right. So see what happens here. So changing the incidence angle changes the outcoming light until it reaches the critical angle or IC. Okay, and at this point, all the incoming light reflects the circular prism, and this is called total internal reflection. This you know. Now the surface is coated with a thin film of noble metal, usually gold, and gold is used in the sensor chip because it is favorable. It has favorable SPR characteristics with stability and a high level of inertness in the biomolecular interaction context. Now at a certain combination of angle of incidence and wavelength, the incident light excites plasmons. Now I was telling you that what is plasmon? Plasmon means that why is gold used suddenly? Because gold has a lot of electrons on its outer surface. It's a very good conductor. It is extremely inert. So that means that it is made up of a lot of electrons and there are a lot of free electrons on its surface. And when this plane polarized light comes and excites these electrons. Now especially plane polarized light is required because this has to come and excite the electrons, and the excitation should be such that it should be such that these electrons get excited and they travel. Okay, and these electrons are called plasmons. Now see what is happening over here. Yeah. So at a certain combination of angle of incidence and energy, that is wavelength, the incident light excites the plasma, that is electron density waves in the gold films. As a result, the characteristic absorption of the energy via the evanescent wave field occurs and SPR is seen as a drop in the intensity of the reflected light as shown in figure 1. So this is the figure 1 that you have, and there it is showing some drop in this intensity. Okay, so in the SPR experiment, one molecule, that is a ligand, is immobilized on the sensor chip and binding to the second molecule, which is your analyte, and it is measured under flow. So response is measured in the SPR units RU. So RU is resonance unit and it is proportional to the mass of the surface, and for given interactant, the response is proportional to the number of molecules bound to the surface. So response is recorded and displayed as a sensogram in real time. Now if you look at this, okay, so next we have ITC. Now here there is one more interesting thing, and that is the sensogram. So initially it is binding and then it is reaching saturation, then there is this kind of a dip and then it decreases. Okay, so as you keep on increasing the volume of the ligand molecule, no sorry, the analyte, as you keep on increasing the volume of the analyte, and then when you are washing it, so that is how the chromatogram goes. Okay, so you can read about this in details. This is very important for your exam point of view. All of these different techniques you must be very well versed with, and you can also search for the questions that are coming into your mind. I will also search for them, so in the next class we can discuss. Okay, right, so now we go to another technique, which is isothermal titration calorimetry. Now here what happens, you're taking the sample and you're heating it. So it is used to measure the heat released or absorbed during a biomolecular reaction. by titrating a solution of protein with a ligand. So there is this protein and there is ligand that you are adding, and it is measuring the amount of heat that is exchanged or it is absorbed. Now researchers can determine the thermodynamic parameters such as binding affinity, stoichiometry and the enthalpy of binding. Okay. Now ITC is one of the very important techniques used to determine label-free. This is also a label-free, highly specific interaction technique between molecules dissolved in aqueous solution, such as protein ligand binding, protein-protein binding, DNA-protein binding, protein-carbohydrate binding, protein-lipid and antigen-antibody interaction. Now protein DNA is very difficult because for ITC you require a lot of DNA and DNA is very expensive. Now, okay, so it is a highly sensitive technique that works by titrating one binding reagent, that is titrant, generally a small chemical compound called the ligand, into the titrant, and usually protein at a constant temperature. This is the titrate and this is the titrant. Okay. Now this technique is commonly used for drug development as it provides valuable thermodynamic information about the interaction of the molecule with its target protein. ITC can precisely determine Gibbs free energy, enthalpy, entropy, heat capacity changes associated with the binding or interaction of the molecule. So now there is this sample cell and there is this reference cell, and the protein of interest is there, and into the protein of interest you are putting in the titrant or the ligand, and then the changes in the heat that are being recorded. So see, the data that you get comes in this form. So ITC instrument measures the amount of power in microcals per second required to maintain a constant temperature difference that is close to zero between the sample and reference cells. You saw two cells, right? There was this reference cell, there was this sample cell. Initially both of them are at the same temperature. Then when you are adding in the analyte molecule, the titrant, into this protein, then there is of course some kind of interactions going on, some breaking of bond and making of bond, all of that leading to enthalpic changes and some kind of displacement, and that is resulting in entropic changes. Because of that, the temperature, there is either heat released or heat absorbed. Because of that, this temperature changes. Now how much heat is required to ensure that this temperature and this temperature of the reference and the sample cell, these are same. So that is recorded. Now initially, the feedback system continuously applies a small power to the sample cell which determines the baseline level. Each injection of the syringe solution, usually termed as ligand, triggers the binding reaction, and depending on the binding affinity, the concentration of the reactants, macromolecules and ligands in the cell, a certain amount of macromolecule ligand complex is formed. The formation of the complex is associated with release of energy or absorption of heat, that is exothermic or endothermic reaction respectively, and then the feedback either lowers or raises the thermal power applied to compensate for such temperature imbalance. Okay. After each injection, the system reaches equilibrium and the temperature balance is restored. Therefore, the recorded signal shows a typical deflection pattern in the form of a peak. This is the deflection pattern that you are seeing with respect to time, okay, in the form of a peak, and then integrating the area under the peak, assuming that the baseline is the reference, provides the amount of heat associated with the injection. As the reactant in the cell becomes saturated, the heat signal diminishes until only the background heat is there. So in a properly designed typical ITC experiment, the binding affinity or dissociation constant or KD and stoichiometry, enthalpy, entropy, everything can be calculated. From this data, Gibbs free energy, entropy change, etc. may also be calculated. Okay. Now there is one more technique. This is fluorescence anisotropy. Now here, this is the difference is that this is not a label-free technique. This does not use heat. Okay, and here the sample can be reused. Okay, so since it is not using heat or anything, the sample is not getting damaged. Now see, fluorescence spectroscopy can be employed to study protein-ligand interactions using fluorescently labeled proteins or ligands. Now, by monitoring changes in the fluorescence intensity, polarization, or lifetime upon ligand binding. So that means the time, the k on and k off rate, etc. Researchers can determine binding affinity, kinetics, and conformational changes in the protein. So once again, see here also polarized light is used. Now you can find out why that is so. Initially there is this depolarized light, and then it is going through a polarizer, then there is polarized light. A polarized light is made to hit the sample. Now what happens is, in the presence of this polarized light, the sample tumbles, it moves, and if the sample, it is initially tumbling, let's say at this particular rate. Now if it is forming a complex with another protein, then it will tumble slowly. So if it is tumbling very fast, then it is giving depolarized light. So initially the light was in this way, and the protein is tumbling very fast because it is a free protein tumbling fast, and this is the light that you are getting. Okay, so this is depolarized. But if the protein is very slowly tumbling, then within the interval that you are measuring the activity, what happens is it is tumbling so slowly because there is a complex. It is now a big complex. It is tumbling so slowly that if you are giving light in this way, it comes out in this way again. So that is polarized emission. So it is not able to depolarize the light or bend the light. Okay. Now, principles of using fluorescence and anisotropy to measure protein-ligand interaction. Free fluorescent ligands or peptides are irradiated with plain polarized light, rotate quickly and emit light. So if it is a very small molecule, if it's a small peptide, it will quickly rotate and emit light in a depolarized manner. But fluorescent ligands bound to large protein, there is a big complex, it is taking time to tumble and emit polarized light. The degree of molecular binding can be inferred by measuring the amount of polarized emission. Okay. Now this is a very simple concept. I hope you have understood. Now we come to EMSA. Now EMSA is something that we repeatedly do in our lab, and this image is also from one of my experiments. Now this is a DNA protein binding experiment. So in EMSA what happens is, there is this protein. This protein is binding to this RNA. Okay, RNA is represented by R. Now see, the binding, the rate of binding is called K on, and the rate of dissociation is the K off. And Kd is equal to K off by K on. Okay, so Kd, that is the dissociation constant, is K off by K on. So if you look at this figure, you see that free mRNA is there. So equal amount of mRNA is added in all the different lanes, and at the second lane from the right, you see that protein has been added. Now because the protein has been added, and of course protein is bigger compared to mRNA, what happens is it will travel slowly in the gel, but your mRNA is traveling very fast. Now mRNA that is bound to the protein, this is also going to travel slowly. That is why there is this shift. Now there is decreasing concentration of protein. So the protein that we add, this is half diluted. That means initially if it was 7 micromolar concentration, the next one will be 3.5, the next lane will be 1.75, next lane will be like 0.8, like that. Okay. So as the concentration of the protein decreases, more of the free mRNA you will be seeing, and this will be running at the rate at which normally free mRNA runs. Okay, the one on the extreme right, the lane on the extreme right is actually free mRNA. Okay, so P total, that the total concentration of the protein is equal to amount of free protein plus amount of bound protein. Now you have no idea about the amount of free protein and the amount of bound protein, right? You only know how much protein you have added, and you only know how much RNA you have added. That is all you know. So from this equation, how do we come to something that we know? Okay, so R total is also R free and R bound. Okay, now fraction bound is equal to concentration of R into P, that is the complex, divided by free R plus complex, and fraction bound is also equal to free protein by free protein plus Kd. So that is how I come to some terms that I know, and from there I can calculate the Kd. So fraction bound becomes P total divided by P total plus KD, and KD is equal to R free into P free divided by complex. Now I do not know R free and P free, I don't know, but I know P total and I will be able to calculate KD from there. Okay. Now how am I going to get this fraction bound? It's very simple. If you look at this lane, if you look at the one on extreme right, that is your completely free, right? And if you look at the one, let's say where that is the third lane from the right, that is where the KD is coming, right? So then for that, the amount of bound protein divided by total amount of RNA. So amount of bound RNA divided by total amount of RNA, that is the bound fraction. So bound RNA divided by total free RNA in lane 1, that is giving me bound fraction. So I know bound fraction, I know total protein, that is how I can calculate the KD. Okay. Now there is this question on surface plasmon resonance. You can try to answer this. In surface plasmon resonance, the sensogram data from a binding interaction provides data on. The first one is binding affinity, rate of association and dissociation, enthalpy of binding, entropy of binding. Which all do you think can be interpreted from SPR data, sensogram? One of the options are anyway getting eliminated, that we can very clearly see from here. Try to answer A. Okay, let's see. Okay, now the right answer is actually D. Why? Because in sensogram data, it can provide information about K on, K off. KD can be determined from K off by K on, but it can't provide information about enthalpy, entropy associated with binding. Okay. If we go back to this, yeah, so it's given here. So binding affinity and kinetics. That is what you get information about. Okay. We go back to the question. Now we have the next question. So next question is about differential scanning calorimetry. This is also a different technique in which protein-ligand interaction can be measured. So regarding differential scanning calorimetry, the question is which of the following parameters can be calculated using a differential scanning calorimetry? So binding constant, now since I have not explained this in this slide, you can read about it. Okay. So whether it is binding constant, stoichiometry of the ligand at excess ligand concentration, and simultaneous ligand binding to several centers even at low affinity. So which of the following do you think can be found out using differential scanning calorimetry? You can read about this. I will not expect you to answer this question because we did not discuss about this in this class yet. Okay, so DSC can measure binding constant, stoichiometry even at low affinity for multiple binding sites. So all the three can be known from differential scanning calorimetry. Okay. Now this is another question on ITC. This kind of questions you can expect, where they'll give you a sensogram and they will ask you to find out what you're observing from it. Okay. So in an ITC experiment, the observed heat changes per injection is fitted to a theoretical fitting curve. So this sigmoidal curve is what is expected, and all the points that you have got, these you have plotted. Now you're getting this. This is called a fitting. Okay, based on your expected values and your experimental values. Now the value of the ligand binding affinity, enthalpy change respectively are. So how are you going to get that? You're going to look at the different units. So based on the units, you're going to find out. Okay, so the first one is time, and this is microjoules per second. And the next one you see is kilojoules per mole, that is heats, the thing for heat, unit for heat, and then there is molar ratio. So binding affinity and enthalpy change can be calculated. So we can look at the answer. Now the explanation is given here. See, this is an ITC plot of 1:2 protein is to ligand binding. Why? Because at midway the molar ratio is 2. So exactly at the midpoint the molar ratio from the second curve, that is the S-shaped curve, is coming as 2. Right? So there is 1:2 protein is to ligand binding. Affinity is around 7 kilojoules per mole, that is halfway of the equilibrium is 7. If you look at that same curve, okay, shaped curve, and the enthalpy change is the total energy released up to the equilibrium, that is 12.5 kilojoules per mole. That is the answer. So this is the one that we are having here, 12.5, and 7 is the midpoint here, and molar ratio 2 is this molar ratio that is telling you that it is a 1:2 binding. Now we come to the last question for today. Now this is a very interesting concept. This is called SASA. So what is SASA? It is solvent accessible surface area. Okay. Now what does that mean? Imagine that this is a protein. It's a normal globular protein. And you throw this in water. Now you expect that if this is the protein, from here this is the midpoint, this should be its radius, right? Normally this is what we are considering as radius. But interesting, when the protein is dipped into water, what happens is this is no longer its radius. Rather, this protein is interacting with small water molecules all over, and the distance between the center of the protein and the center of the water molecule that it is interacting with, this becomes the new radius called the hydrodynamic radius. Okay, and the surface area of the protein that is exposed to these water molecules, these are called solvent accessible surface area. Now if you look at this figure here, you will see that this entire region surrounding the protein, this is actually the radius of the water molecule plus the radius of the protein. Together that makes up the total hydrodynamic radius. Okay, now why is this very important? Because this tells you a lot about the interactions of the protein. Okay, so based on the solvent accessible surface area, you can find out which regions in the protein they are involved in protein ligand interactions. Now, the question is, which among the following cannot be obtained from the solvent accessible surface area? One is protein ligand binding strength, hydrophobic free energy of transfer from polar to hydrophobic environment. Third is the interface area due to change of surface area upon protein-protein or protein ligand complex formation. Okay, so initially this was the protein, but let's say that another protein comes and binds to it, or another ligand molecule comes and binds to it. Then what happens? Definitely the water molecules that were here in this region of the protein, they get displaced and they get rearranged around this protein, right? So there is change in this solvent accessible surface area now. So the interface area due to change in solvent accessible surface area upon ligand or protein complex formation, that definitely you can know. And what else can you? So that is actually the interface, binding interface. Okay. And the region in the protein that is there without the water molecule, that is called the solvent excluded surface. Okay. So if we look at the answer, so the answer is B. So all of these could be obtained from the change in the solvent accessible surface area. So all of these different things can be obtained from the solvent accessible surface area. Okay. Right. So that was it for today's class. All the materials are provided here. So all the different books that I have referred to, the different experiments from virtual labs and different websites that I have referred to are mentioned here. Thank you for your time. Thank you for your patience. If you don't have any more questions, we can end today's class and we'll meet again next week. Thank you so much on Tuesday. Thank you so much. Okay. So then I'm ending the call. Okay. Thank you so much for your time.